In this class, we'll try to learn about GI motility. The learning objectives of this class would be correlate electrophysiology of GI smooth muscle with GI motilities, draw a label diagram of slow wave of GI smooth muscle, understand the relation between electrical and mechanical response, outline the organization and function of enteric motor neurons, name the GI motilities and give their functions, mention various GI sphincters and give their function, appreciate applied aspects of physiology of GI motility. Physiology of smooth muscles. Movements of GI tract assist in propelling foodstuffs in the forward direction, breaking down the foodstuffs into smaller particles and mixing of the food with the GI secretion for proper digestion and absorption. This is possible due to the presence of smooth muscle in the wall of GI tract and the presence of developed system of enteric motor neurons. So here are some of the specialities of the GI smooth muscles. GI smooth muscles are unitary type of smooth muscles. The unique feature of GI smooth muscle is that contract spontaneously without external neural and humoral influences. They also contract when stretched without external innervation. There is no structured neuromuscular junction. The GI smooth muscles are long about 500 mm and slender around 5 to 20 mm in width. They are arranged in bundles. Electrophysiology of GI smooth muscles. The type of couplings in smooth muscle can be electromechanical coupling. Like in skeletal muscles, smooth muscles have electromechanical coupling. That means depolarization of the membrane causes opening of the voltage gated calcium channels and increases cytosolic calcium, which leads to muscle contraction. So there can be pharmacochemical coupling. So in addition, smooth muscles have an additional mechanism of pharmacomechanical coupling in which binding of a ligand to its receptor on the muscle membrane leads to opening of the calcium channels and increased cytosolic calcium without change in the membrane potential. So there can also be presence of mechanical coupling. The stretch of the smooth muscle causes muscle contraction, so which is known as mechanical coupling. The stretch of the muscle opens stretch sensitive calcium channel on the smooth uh, on the muscle cell membrane. Calcium influx causes muscle contraction. Syncytial tissues and pacemaker tissues. GI muscles have numerous gap junctions that easily transmit electrical impulse from one cell to another cell. This accounts for the syncytial nature of smooth muscle in GI tract. There are also pacemaker tissues in the GI tract and generate and spread the electrical impulses. Basic electrical rhythm and slow waves. The resting membrane potential of the smooth muscle of the GI tract exhibit a wide range of fluctuation compared to other type of muscle fibers. The electrical slow waves are wide, spontaneous, rhythmic fluctuation in the membrane potential ranging between minus 65 to minus 40 millivolts. The slow waves, which oscillate significantly, are called as basal electrical rhythm, that is BER, that occurs due to the wide variation in the RMP of GI smooth muscles. The BER present in all parts of the GIT except in the esophagus. Slow waves in many regions of GI tract represent only as a BER, whereas other places like gastric antrum and intestine they trigger action potential. Slow waves are generated by the interstitial cells of Kajal located between the longitudinal and circular muscle layers. So here the picture showing electrical slow waves of the small muscles where the membrane potential ranges between minus 60 to minus 30. Interstitial cells have long processes that form gap junctions with the longitudinal and circular smooth muscles. This enables easy and rapid conduction of slow waves from interstitial cells into the smooth muscles. The slow waves spread throughout the smooth muscles of each segment of GI tract. There are different phases of slow waves. A typical slow wave has a four phases. Phase one is the rising phase, that is this phase of depolarization, which occurs due to the opening of voltage gated calcium channels. Phase 2 is the plateau phase that occurs due to the balance between inward calcium current and outward potassium current, that is potassium efflux. Phase 3 is the falling phase, that is repolarization, occurring due to the activation of voltage gated potassium channels. And phase 4 is a return to the membrane potential. So these are the four phases. The phase one is depolarization. Phase this phase is plateau phase, that is repolarization, and coming back to the normal resting membrane potential. So these are the electrical waves of the small intestine. The frequency of the slow waves is 3 per minute in stomach, 6 to 10 per minute in colon, 15 per minute in jejunum and ileum, and per minute 18 per minute in duodenum. The amplitude and frequency of slow waves are influenced by activity of extrinsic and intrinsic nerves and by the hormones. Usually sympathetic stimulation decreases the amplitude and parasympathetic stimulation increases the amplitude of slow waves. Here are the showing the frequency of the slow waves in stomach, the colon and jejunum, where it is 15 contractions per minute, waves per minute. Sorry. Action potential. When peak of slow wave exceeds the threshold, the action potential is triggered from the peak. These action potential facilitate the force of contraction of the smooth muscle. Action potentials have a longer duration, 10 to 20 milliseconds, and usually they do not overshoot. The rising phase of the action potential is caused by influx of calcium and sodium. The repolarization is due to the potassium efflux. The magnitude and frequency of action potential are enhanced by vagal stimulation and inhibited by sympathetic stimulation. So this is the diagram showing the action potential in the GI muscle. Note that once the threshold is reached, the action potential is generated on the peak of the slow wave. So this is called as spike wave. The relation between electrical and mechanical responses. Slow waves that are not accompanied by action potential, they do not excite, they do not elicit the contraction, whereas slow waves that are accompanied by action potential evoke contraction. The greater the frequency of action potential that occur at the peak of the slow waves, the more intense is the contraction. Electrical coupling between cells. The smooth muscle cells of the GIT are well coupled or better coordinated. The electrical changes spread rapidly from one cell to another cell. However, the electrical coupling between circular cells is better than the longitudinal cells because of this. Circular muscles have more number of gap junctions. So this diagram showing the electrical and mechanical responses of the GI tract, where the number of the spikes are more or action potentials are more, the amplitude of contraction increases. With only the slow wave, there is no contraction. With less number of spike potential, there is a less amplitude of contraction. Enteric motor neurons innervate the smooth muscles of GI tract and are the final pathway for output of the information from ANS and enteric nervous system to the muscle cells. They neither form a regular neuromuscular junction nor do they release neurotransmitter at their axon terminal. Rather, most of the motor axons release transmitters from varicosities that occur along their axon during the propagation of action potential. And the neurotransmitter diffuses to reach the muscle or the interstitium. The major excitatory neurotransmitters are acetylcholine and substance P, and the inhibitory neurotransmitters are catecholamines ATP, VIP, and nitric oxide. Cell bodies of excitatory motor neurons are present in the myenteric plexus, and the axons project arboreally to innervate the muscle fibers. There are also secretomotor neurons that stimulate secretion of water, electrolytes, and mucus from GI tract. The cell bodies of secretomotor neurons are present in the submucosal plexus. Stimulation of the secretomotor neurons release histamine during allergic reaction and produce neurogenic secretory diarrhea during stress. So this how the enteric nervous system is going to uh, work, and the enteric nervous system is the final common pathway for GI effector organs. Okay, so central nervous system is going to affect the autonomic nervous system, which indirectly affects on the enteric nervous system through the interneurons or motor neurons. Um, so motor neurons, they release neurotransmitter whether excitatory or inhibitory, so which are going to act upon the effector organs. They act on muscle secretory cells and blood vessels, and affects the motility, secretion, and blood flow to the GI tract. Basic patterns of GI motility. The first one is peristalsis. So this is a reflex response to GI tract to stretch, which results in organized propulsion of luminal content of gut in a forward direction. This response is present in all parts of GIT starting from esophagus to rectum. When the gut wall is stretched by contents of its lumen, peristalsis is initiated. Mechanism. Stretch initiates the circular ring like contraction behind the stimulus, that is the part before the area of distension, and relaxation in front of it. The part behind the stimulus is called as propulsive segment as it propels the bolus of the food or chyme, and the part ahead of the stimulus is called as receiving segment and it receives the bolus. In the propulsive segment, the circular muscles contract and longitudinal muscles relax. In the receiving segment, the circular muscles relax and longitudinal muscles contract. The contraction wave then moves in a forward direction, that is from oral to anal direction. This helps in propelling the contents aborally. Role of cholinergic neurons. Cholinergic neurons that present in retrograde direction with cell bodies towards anus and terminal towards the oral cavity activate the neurons that release substance P and acetylcholine. These neurons stimulate circular muscle contraction and help in the formation and progression of the contractile ring behind the stimulus. So this diagram showing the propulsive uh, movement, okay, where the foot bolus, it causes the contraction of the circular muscles behind and relaxation of the longitudinal muscles, whereas in the distal segment, in the receiving segment, it causes the relaxation of the circular muscle and the contraction of the longitudinal muscles, and which is going to help in the movement of the bolus in forward direction. Cholinergic neurons that present in antegrade direction, cell bodies towards oral cavity and terminal towards anus activate the neurons that secrete VIP and nitric oxide. These neurons assist in relaxation of the circular muscle in the portion of the gut ahead of the stimulus. Factors influencing peristalsis. The normal rate of transmission of peristaltic wave varies in different parts of the GI tract, ranging between 2 to 12 centimeters per second. This is influenced by intrinsic and extrinsic nerve supply. Sympathetic stimulation inhibits peristalsis and parasympathetic stimulation enhances it. Though the transmission of peristalsis is affected by autonomic influence, its genesis is independent on it. It is affected by hormones secreted locally. Serotonin secreted by local stretch of gut enhances the peristalsis. Migrating motor complexes. During interdigestive period, During interdigestive period, the electrochemical activity of GI smooth muscle is altered. A cycle of motor activity migrates from stomach to distal ileum. So this is called as migratory motor complex or MMC or long peristalsis. There are different phases of MMCs, that is phase 1, phase 2 and phase 3. Phase 1 is a quiescent phase. Phase 2 is a period of irregular electrical activity and mechanical activity. Phase 3 is a period of burst of electrical activities. Usually it originates in the stomach and transmitted aborally at a rate of 5 centimeters per minute. It occurs at interval of 90 minutes, after which the cycle is repeated. Significance. MMC occurs in the interdigestive phase. The phase 3 of MMC results in long peristalsis, which starts from stomach and terminates in the distal part of the ileum. Therefore, it sweeps the gastric and intestinal contents into large intestine. Consequently, MMC is alternatively called as sweeper of the GI tract or housekeeper of the GI tract. It clears the stomach and intestine of their luminal content so that they are ready to receive and digest the next meal whenever ingested. Characteristically, MMC immediately stops when food is ingested. Retroperistalsis, usually peristalsis moves in a forward direction. However, there is an obstruction in the lumen. Reverse peristalsis can occur, and this is called as retroperistalsis. Vomiting occurs due to retroperistalsis. Retroperistalsis is also initiated by emotional factors or due to stimulation of medullary vomiting center. Segmentation contraction. This commonly occurs in the small intestine. So it is characterized by closely spaced contraction of the circular muscle layer. These contractions divide the small intestine into mini segments, therefore, the movement is called as segmentation. It is primarily meant to mix chyme with the pancreatic and intestinal secretion and also bring the fresh chyme into contact with the mucosal surface. Therefore, segmentation is also called as mixing type of movement. Role of sphincters. Sphincters are rings of smooth muscles that remain in a stage of continuous contraction. So these are anatomical and physiological sphincters. The upper esophageal sphincter is an example for physiological sphincter, which prevents esophageal contents to regurgitate back into oral cavity. The anatomical sphincters are located at gastroesophageal, gastroduodenal, and ileocecal junctions, and at the opening of the bile duct in the duodenum and the opening of the rectum into anus. Thus, sphincters uh, separate the two specialized compartments. They regulate the movement of the content from the upper compartment to the lower compartment and prevent backward movement of contents from lower to upper compartment. So, this is in brief about uh, the motility of the GI tract. Thank you.